Hello, this video is the second video covering the topics in the muscle tissue chapter in an A&P course. So the first video covering the first part of the chapter focused on naming all of the structures in muscle tissue, but then also on the sliding filament theory, the idea that there is a functional unit in muscle cells known as the sarcomere, and that it is the interaction between the proteins in the sarcomeres, which then cause sarcomeres to contract as myosin filaments pull on actin. And then because myofibrils are made of sarcomeres and because muscle cells are made of myofibrils, that the contraction of the sarcomere then explains how the entire muscle contracts. So that was the major topic in the first part of the video. This second part covers a number of topics, a couple of major ones and then a handful of shorter ones. Now, two of the topics I'd like to address now, the action potential and the neuromuscular junction. Well, they're a problem. And so when I teach, this is how I teach it. I tell my class that in this chapter on muscle tissue, there are three big concepts, essay-worthy, exam questions. There is the sliding filament theory, there are action potentials, and then there are synapses. And that's a lot for one chapter, and it would overwhelm students to spend an hour on this one, an hour on that one, an hour on that one. It's just a lot. But two of those three topics, the action potential and synapses, are just as, if not more appropriate to cover with the nervous system, which in Anatomy and Physiology 1 is the topic which comes after the muscular system. And so therefore, when I teach this chapter, I cover two of those topics, in a summary fashion, just an introduction, with the idea that in the nervous system, the action potential is given its due hour-long treatment, and synapses are given a more thorough treatment. And so these topics appear in this chapter, but they appear to a greater degree in later chapters, so then I will cover them in a cursory fashion here. So, cells conduct electricity. They can. Nerve cells do and muscle cells do. Muscle cells are excitable. That's a special characteristic. Most cells don't conduct electricity. And they do so, as will be discussed in much greater detail later, as there is this wave of depolarization as proteins, which let ions into the cell, open. And so that as positive ions come into the cell here, and then at the next place, and the next place, and the next place, a wave of electrical change passes over cell membranes. So it's called an action potential. Action potentials can pass along the membranes of both nerve cells and muscle cells. But when they reach the place where cells touch, what's called a synapse, in most cases, not all, in most cases, the electrical action potential cannot jump from cell number 1 to cell number 2. And so therefore, here the message is then converted, instead of being an electrical message which travels along one cell, to a chemical message which now jumps from one cell to the next. So at a synapse where one cell touches another, we're going to have chemicals called neurotransmitters pass, which then transmit the message. There are different types of synapses. So there are synapses where nerve cells touch each other, interneuronal junctions. There are neuroglandular junctions. But what we'd like to focus on here is a neuromuscular junction, where cell number 1, before the synapse, what we could call the presynaptic cell, that's a neuron, and cell number 2, what we call what comes after the synapse, what we could call the postsynaptic cell, that is a muscle cell. And so what's going to happen, and once again, this is the very cursory treatment, with the idea that this is covered in greater detail elsewhere, is that the electrical action potential, which reaches the end of a neuron, called the synaptic knob, will cause some of these bags of neurotransmitter signal. They're called vesicles. Some of these vesicles will then undergo exocytosis, so that the chemical message of neurotransmitters is then put into this space, what's called the synaptic cleft between the presynaptic neuron and the postsynaptic muscle. And the muscle knows that this message is coming. All right, so it's not a surprise that this neurotransmitter comes here. And so therefore the muscle cell is making receptors for that neurotransmitter. When the neurotransmitter then binds to the receptors, then that causes this cell membrane to depolarize and start that action potential electrical message, which will be covered in greater detail later. And so now what was an electrical message traveling on cell number 1 has been continued as an electrical message, which will spread along cell number 2, the muscle cell, because the neurotransmitter crossed the synaptic cleft and it stimulated that muscle. So the neuron is going to secrete these signals, neurotransmitters, which then enter this synaptic cleft and then reach the muscle cell. And if you look under a microscope, these dark lines, these are the nerve cell axons, which end in synaptic knobs. And then the redder cells, and you can see striations, those are the muscle cells. So you can see that in muscle tissue, there are ample nerve cells. Look at all of these nerve cells, which then reach out and touch the muscle cells and form these neuromuscular junctions. And so I always, joke with, my students where, are you scared that, when you're on the dance floor at the anatomy and physiology party, that all of a sudden your arms, start flailing about and start hitting your dance partner, or when you're dissecting in lab, that all of a sudden the hand waving the scalpel, starts flailing around. No, because these muscle contractions are under the control of the nervous system. The nervous system tells them what to do. And so it is electrical messages on the nerve cells, which then are converted at a synapse to electrical messages which travel along the muscle cells. And once again, these neuromuscular junctions will be covered later, but here you see once again the nerve cells. They are the ones which will control the contractions of muscles. And as I'll mention again in just a second, this is a video which is coming up. Each nerve cell will then branch out and touch a specific number of muscle cells. When the nerve cell has its electrical message, its action potential, then all of the muscle cells that it touches go to action potential, and they then contract. It's what's called a motor unit, as we will see. Okay. If you were to say be asked, how does the message of a nerve cell then end up in the contraction of the muscle, the very quick summary description is that the action potential spreads over the nerve cell, then is converted to a chemical message at the synapse, which then will start an electrical message on the muscle cell, all right, because the muscle cell was waiting with receptors. And then going back to the anatomical terms in the first half of this chapter's videos, now the action potential will spread over the sarcolemma. That is the excitable cell membrane of the muscle cell called the sarcolemma. So the action potential then spreads over the sarcolemma, then through the T tubules. These are these channels which allow the electrical message to go deep inside the cell, to the sarcoplasmic reticulum, which is storing calcium. When the sarcoplasmic reticulum is then signaled with this electrical message, it releases its calcium. And as we know from the first half of the video, the calcium binds to troponin, moves tropomyosin out of the way, and now the myosin heads can pull on actin. And this then causes the contraction of the sarcomeres, which results in the contraction of the entire muscle cell. Now, because that was such, an important concept, let's just kind of repeat it one or two more times. So once again, there is a connection between the nerve cell electricity and the contraction of muscle. It's what's called the excitation-contraction coupling. And so the action potential of nerve cells then cause an electrical action potential to spread over the sarcolemma of a muscle cell. This action potential then goes from the sarcolemma through the T tubules down deep into the cell to where the sarcoplasmic reticulum is. The sarcoplasmic reticulum then releases calcium, and then the calcium, as we know, binds to troponin, moves tropomyosin out of the way, and now all of a sudden the active sites of actin are open so that the myosin proteins can attach, pivot, detach, return, attach, pivot, detach, return. As we will discuss in a future chapter, how is it that electricity can do this? Well, there are ion gates like those which would release calcium, which are voltage regulated. And so when an electrical message reaches them, it changes the voltage, and then they go from being open, from closed to open, and then calcium is released. And then, as I had said, the active sites of actin are now open, and then the sarcomeres can then contract. So once again, there is an excitation-contraction coupling where the electrical messages which travel over cell membranes are then coupled to the contraction of sarcomeres thanks to those muscle proteins. So just one more time, once again, it is an important concept that the electrical message goes over the sarcolemma through the T tubules, and here this sarcoplasmic reticulum has stored calcium. There are voltage-regulated ion gates. When the electrical potential changes, the calcium is released. Once calcium is released, the active sites of actin, these darker blue spots, are no longer covered by tropomyosin because calcium binds to troponin, which moves tropomyosin out of the way. The active sites are now open, and the myosin heads can now attach, pivot, detach, return, and this results in the contraction of sarcomeres. And since muscle cells are made of sarcomeres, this then results in the contraction of muscle. So I know it's odd to talk about electricity in the body. I mean, we often think that, electricity and the body are two things that don't go together. I could die. The answer is, well, no. I mean, our thoughts are electrical. The commands for muscle cells to move are electrical. And so whether you had an ECG, which is measuring the electrical activity in the cardiac muscle of the heart, as you can see here, an EEG, an electroencephalogram, then you can then measure the electrical activity that occurs in muscle cells. Before we get into another big topic, here is a quick definition. Once again, muscles technically don't contract. Now, it's a silly little definition, because what's defining the contraction? If I tell my biceps to contract, will they? Well, that depends. If I have nothing in my hand, there's just air then of course the contraction of this muscle can result in a shortening. Well, let's say I'm trying to lift something very heavy, all right? Then I can say muscles contract. They won't though, all right? Because if it's too heavy, then I can't flex my forearm because it's too heavy. And so technically, what muscle proteins do is they generate tension. And so sometimes you can have what's called an isometric contraction where force is generated but muscles don't shorten. In what's called, and so once again, what's determining whether the muscles shorten or not is actually the comparison between the weight of the load and the tension that you are creating. If you don't create enough tension to overcome the resisting force, then your muscles won't shorten. So in contrast, an isotonic contraction, the force exceeds the resistance, allowing the muscle to shorten. So if I were trying to lift small things, then I could, and muscles would shorten because the force that I was generating exceeds that. And so just, even though it's easy to say muscles contract, technically they produce tension, and this then can result in either isometric contractions that are the same length. The muscle actually doesn't contract, it just generates force, or an isotonic contraction. Now, the amount of force can be changed through three ways. All right. So here's the thing: if I were to lift something, if I were to lift, just something very, very light, this notepad, I can do it. If I were to try to lift something much heavier, I could. So the same muscle, the biceps brachii, can generate different degrees of force. So how is that possible? How can we use the same muscle in different ways, generating different degrees of force? And so one of the ways that we can change the amount of force is that we can change the number of motor units. So just to define, here comes a nerve cell. It is going to send a muscle cell to tell a muscle cell to contract. But even though this nerve cell will branch, it doesn't touch every muscle cell in this muscle, say the biceps. Let's say this neuron only touches 3. This one only touches 1. This one touches 3. This one touches 5. This one touches 4, etc. It can vary. So when this nerve cell carries an electrical message, an action potential, the only muscle cells which respond are those muscle cells which it touches. So this neuron is not going to make these cells contract. When it goes to action potential, these cells don't know that. They don't touch it. So a neuron has to synapse with these muscle cells. So when that nerve cell goes to action potential, these muscle cells then contract. And then we'll see that in the other ones as well. So when each one undergoes a synapse, and I'm sorry, once I'm zooming and showing videos, it's a little jumpier than the actual video is, then only some of these muscles contract. So this is what is known as a motor unit. All of the muscle cells that a neuron touches are called its motor unit. And when that neuron goes to action potential, only the cells of the motor unit respond. All right, so that's just what a motor unit is. Now, I can change the amount of force that I'm using in lifting an object, for example, by changing the number of motor units that I'm using. If I use one motor unit, then a couple muscle cells will contract. But then I could use 2 motor units. I could use 3 motor units. I could use most of the motor units. I could use all of the motor units. And so therefore, the amount of force that I'm generating in a muscle when I'm lifting something can vary. So if I'm lifting something light, I could just have a couple of motor units respond. If I'm trying to lift something heavier, more motor units could be used. So that's one of the 3 ways that I can vary the amount of force that I generate. I can change the number of motor units that I use. One last thing on motor units is if a motor unit is pretty big, all right, like this one is the biggest, this one's the biggest one here, where 5 cells respond when this nerve cell sends a message. In actuality, if we were to look at, say, the back or the calf, there could be hundreds of nerve cells in a motor unit. In that case, I don't have very specific control of a body part. So for example, in my back, when I tell my back muscles to contract, if a motor unit has 100 muscle cells in it, my choices are tell 100 muscle cells to contract, or 0. 0, 100, nothing in the middle, because once this nerve cell sends a message, all of the cells in the motor unit respond. So where I have motor units with a lot of muscle cells per neuron, I don't have a very specific control of my body. But if we were to look at the fingertips, or the vocal cords, or the eye, and what I often tell my students, here, look at me, and don't move your head, and now change the focus of your view from here, my finger on my nose, to now here, my eye, but don't move your head. And so they move their eyes, and they change the plane of focus here. And then I ask, that was pretty amazing, because seriously, how many eye muscles, how many cells contracted to change your plane of focus from here to here? I don't know, let's say 3, all right? That's a really small number. If you have a big body that's made of 100 trillion cells, and you can send commands so precise that only 3 cells respond, that's pretty impressive, all right? So there are places where motor units are very small. 1 nerve cell touches 3 muscle cells, 2, 1 maybe. All right, so places like our fingertips, our vocal cords, our eye muscles, we have extremely precise control because motor units are small. So that was a side note. The smaller the motor units, the greater the precision of control in that area. We'll see that again when we look at the parts of the brain. So I have a region of the brain that controls muscle contractions. And in this part of my brain, my back takes up like that much space, whereas my cheek, all right, my fingers take up that much space. So if you were to just look at my brain, it looks like my hands are bigger than my back. They're not. But because my motor units are so big in my back, I don't need many neurons going there if each neuron is going to touch hundreds of muscle cells. And so actually, when you get to the brain, you'll say, oh, that makes sense now, going back to those motor units. Okay. But now, the second way that I can change the amount of force that a muscle is generating is I can change how frequently I tell the muscles to contract. So just a couple of quick definitions. 1, I can do what's called a twitch. And in a twitch, I say muscle contracts. The muscle contracts, I let it relax completely, and then now if I want to, I can tell it to contract again. All right, so in a muscle twitch, I tell it to contract, but then I let it relax completely. But I have another option. I could say, I told it to contract. Instead of letting it relax completely, I could now send a second message before it has completely relaxed. So it could start to relax when I hit it with another message, contract again. And so it contracts before it gets to fully relax. So this is what's called wave summation, all right? So I tell it to contract, it contracts, but before it gets to relax completely, it is told a second time. And once again, I apologize that zooming and playing videos is not as seamless as I'd like it to be. So wave summation, the muscle doesn't get to relax completely. One little quick aside thing is, so here it is, look, it started to relax. It did not relax completely when I told it to contract again, all right? Now, one other little side note is that the force that this muscle generates will actually be a little higher now. And the reason is, if I say muscle contract at the sarcomeres, calcium gets released, and then I have to pump it back into the SR. When it gets pumped back into the SR, that ends the contraction. But if I tell the muscle to contract a second time before it is relaxed completely, it releases more calcium, but there's still calcium left over from the previous contraction. And so that the force generated in this second contraction is just a little bit higher than it was in the first one, because in addition to the calcium from the second one, a full dose, I still have some of the calcium left over from the first one. So actually now I'm generating a bit more force with wave summation. I'm summating, I'm adding these, the amount of calcium together. Last one in this one, I ask my students, tetanus, good or bad? And they often, my students say bad. And that's not a wrong answer, but it's not entirely right either. And the reason is because tetanus means two different things. So tetanus is sustained muscle contractions. Sometimes that occurs after an infection, like Clostridium tetani, you can die, all right? Because if your breathing muscles contract and you can't have them relax, you can't breathe anymore. That's bad, you can die. So that tetanus is bad. But if I'm, say, helping someone move a couch, all right, and I'm now lifting something heavy, yes, I could tell my biceps, contract, relax, contract, relax, contract, relax, contract, relax. But after a while, can I just say, just contract and hold it? Just contract, hold it. Well, that's what's called tetanus. And that is not bad. That is a normal part of skeletal muscle contraction and exercise, not cardiac muscle. Cardiac muscle doesn't do tetanus, but you can talk about that later. Skeletal muscle does tetanus commonly during exercise. So tetanus can be used in two scenarios, and one of them is not bad, it is normal. Tetanus is a sustained muscle contraction. So if now, watch the purple action potentials coming in. They come in so frequently that these muscles don't get a chance to relax. They're told to contract, but before they begin to relax, they're told to contract again, and then they're told to contract again. It's like go, 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 and they don't get a chance to relax, and so they just maintain this state of contraction. That's what's called tetanus. And so if we were asking what makes the difference between, you know, lifting something light and the work of lifting something heavy, the second thing is I can change the frequency of stimulation. So by either deciding, hey, my muscles can twitch, they can contract and relax completely, they can undergo wave summation, they can undergo tetanus, I'm altering the force that I'm generating by changing the frequency of stimulation. Then the third and final way that we can change the amount of force generated. Now, I apologize, I'm going to use this video then obviously here. I would try to demonstrate this in a class, but I think I would just be silly if I tried to, you know, back up and do it here. So I say, let's say you're in a gym, you know, the person from the lab table that you're trying to impress, you know, comes in and you decide you're going to try to lift something really, really heavy. All right. Well, how would you grab that? Right. And so I say, well, you wouldn't, you know, hold your arms like this, bend down and try to lift up this weight. I mean, that's a silly, you know, position. You wouldn't stretch your arms out like this and reach down and grab it and try to lift it. That's a silly position. You would hold your arms like this. If you wanted to try to lift something heavy, you would grab it like this. Why? Well, because when you change the length of your, you know, your arm here, you're changing the length of the muscle cells. You know, so here, muscles are shorter that then have to stretch out when you're here, and thus you're changing the length of the sarcomeres. All right, so when you flex and extend your forearm, you're changing the length of the muscles. Now, in the sarcomeres, myosin heads can contact actin. But if you make the sarcomere short, or if you stretch it to its longest, you're actually changing the number of myosin heads, which can access the actin filaments, all right? So just at any specific moment, how many of these red knobs can actually contact actin? If the sarcomere is too short, the number is not at its greatest point. If the sarcomere is stretched out, then maybe these could still, maybe this one could still contact actin, but now this one couldn't if we stretch the blue too far this way. And so we generate the most force in exercise when the most number of these globular heads are contacting actin. But if the sarcomere is shorter than its optimal length, or longer than its optimal length, you reduce the number of these heads, which can actually contact the actin, and thus force is less. And so when I change the amount of force I generate in a muscle contraction, I can do that by changing the number of motor units. The more motor units, the greater the force. I can change the frequency of stimulation. I can undergo tetanus. rather than muscle twitches, at which case in tetanus I'm generating more force, or I could have an optimal length of the sarcomeres where the greatest number of myosin heads is contacting actin. Okay. Now, as I talk about exercise in the upcoming videos, obviously ATP comes into this. So, just quick little video on this. So this is a molecule of ATP, and as you remember from earlier in the course, the third phosphate here. So we make ADP by taking ADP and attaching a third phosphate. That's a high energy bond. It took a lot of energy to create that bond. And so therefore energy is stored there in the same way that if I lift a chair, I take a lot of energy to lift a chair, energy doesn't go away. It's now in the form of potential energy. If I let go of the chair, you can see that energy becomes kinetic again. So just as the position of a chair can store energy, the position of electrons in chemical bonds can store energy. And so there's energy stored here. And if I break that bond, that energy can now be used. And muscle proteins need ATP for this myosin head to detach and then return to the cocked and ready position. So in the cross-bridge cycle of attach, pivot, detach, return, attach, pivot, detach, return, ATP binds to the myosin heads, all right, when it's attached, and after it pivots, then using this energy, it then detaches and returns to the ready position. So just a quick thought, rigor mortis occurs because after death, the sarcoplasmic reticulum starts to break down and releases calcium. And now that calcium is released, muscles can contract and the muscles can become rigid. But in order for the muscles to detach and return, they need ATP. And after death, new ATP is not being generated. And so the muscles contract as the myosin heads attach to actin, but they can't let go. That's why there's this rigidity for a brief time after death, which then ends when these muscle proteins start to break down. And so the energy which is then stored at that bond, when this bond is broken, converting ATP into ADP plus P. This is what ATP is converted into when you break this bond. Then that energy then causes the myosin head to return to its cocked and ready position so that it can do that again. Okay, so we need ATP in muscle contraction for this cross-bridge cycle, which gets us to exercise. How do we get the ATP to then exercise, if you're trying to run or jog or go up a flight of steps, etc.? Well, it turns out this is going to be a long discussion in that we have to then go through ATP is generated in different ways in different phases of exercise. So, there will be multiple answers to this question. First, we do have ATP just waiting in muscle. All right, so if I were to tell my muscles to contract, there's ATP waiting there. They can then use this ATP to generate energy. All right, and so I already have, can I just do this? Okay, I think it repeated. So there's already ATP in, I'm sorry, there's already ATP there. But if I were to say, try to run a 100-yard dash, I do have ATP stores waiting, but this isn't enough for prolonged exercise. It can get me through a couple of seconds of maximal exertion. So if I were to just do maximal exertion, I could, sprint for, a handful of seconds using the ATP that I have in my body. But then after that, all of the ATP in this muscle tissue is now in the form of ADP plus P. So now I have a problem, all right? I either have to stop exercising or find a second source of ATP. So, I make this, silly analysis. Let's say something, is chasing you with sharp teeth, okay? So you start running away from it using your ATP stores. But seconds later, when ATP runs out, you have an option. Do you fall over, which is not a good option with the thing with sharp teeth chasing you, or do you continue? If you continue, it is because you have a second source of ATP. What is that? The second source of ATP is what's called creatine phosphate. All right, so we use up all of our ATP within a couple of seconds. And so now we have ADP. So here's adenosine, so there's adenine, there's ribose, and here's two phosphates, adenosine diphosphate, ADP diphosphate, has two phosphates. You want ATP triphosphate. Well, it turns out that in your muscle tissue, you have what's called creatine phosphate. There's another molecule sitting there derived from an amino acid called creatine, and it's got a phosphate attached to it, and that can be now a phosphate donor. ADP can now go to creatine phosphate, and now then take its third phosphate here. So creatine phosphate plus ADP can undergo a chemical reaction that then becomes creatine without the phosphate because it gives it away. And now you have ATP again. So creatine gave up its phosphate, and now here we have ATP once again. And so now I can keep running. So ATP plus creatine phosphate, which can now regenerate my ATP stores. This lets me run even longer. I can now run for 15 seconds. All right, now that's good. Maybe the 4 to 6, I couldn't have finished a 100-yard dash in that time. All right, but now I can run a 100-yard dash. But if the thing with sharp teeth is still chasing me, well, now after 15 seconds of maximal exertion, I'm out of ATP, I'm out of creatine phosphate, and I have two choices. I either fall over, which when something with sharp teeth is chasing me, not my first option, or I can find a third source of energy. All right, so once again, here, this individual here, is lifting something with muscle. So they're going to start off using their ATP stores, but then they run out of ATP. And so then they use creatine phosphate. This phosphate on creatine can now donate it to ADP to make it ATP again. So ATP is becoming ADP plus P. So it's adenosine diphosphate, but I can take this adenosine diphosphate and then add a third phosphate to it and renew my phosphate stores for a bit. So this is great, but it won't last forever. And so then the question comes up, what now? Well, now the answer is sugar. All right, I'm now going to break down glucose as an energy source in what's called cellular respiration. Now, this is a little awkward in that if you were, say, studying general biology, cellular respiration, this could be a week's worth of lecture. All right, it's a big topic, it's an important topic. In anatomy and physiology too, there is a chapter after the digestive system called metabolism, and half of that chapter is on cellular respiration. So here we are in the muscle chapter in, say, anatomy and physiology 1. And we'd like to understand cellular respiration, but we don't want to spend a week on it the way you would in, say, general biology 1. And we know that the most thorough coverage of this topic occurs in anatomy and physiology 2, where cellular respiration is the first half of the metabolism chapter. So this, remaining, coverage of it will be thorough enough to apply it to muscle and exercise, but not as thorough as it could be, obviously. And so if you would like a more thorough coverage. I mean, not only do I have more videos on this, but that would come, say, in anatomy and physiology 2 or a general biology class, not here. So if I want to take sugar for energy through cellular respiration, I then perform something known as glycolysis. Now, glycolysis, what happens is I take glucose, which is a 6-carbon molecule, and then I essentially break it in half to make 2 3-carbon molecules. I don't require oxygen, so this is an anaerobic process. This occurs in the cytoplasm of the cell, so I don't need the mitochondria, a special part of the cell. And I get a little bit of energy, 2 ATP. So I need ATP to keep running, because once again, my option are, fall over with something with sharp teeth chasing me. That's not a good, option. And so glycolysis, where I break glucose in half into 2 pyruvates, that's a good option, and I can keep running. All right. So this is good for now. So here's just a quick image of covering glycolysis in greater depth. So here's sugar that I ate. It was in a potato chip or a French fry or in corn or in rice or in noodles or in bread or in cake or something. We eat a lot of glucose because that's what starch is made of. It's a 6-carbon molecule. There's energy there. How do I get the energy there out? Well, all of the following chemical reactions are occurring in what's known as glycolysis. Once again, you could spend a long time on glycolysis, but in essence, what's happening here is you're taking the 6-carbon molecule of glucose and you're breaking it in half, all right? And the 2 halves then are what are called pyruvate, all right? So pyruvate's a 3-carbon molecule, and in the process of glycolysis, which occurs in the cytoplasm, without oxygen, we can make pyruvate. Great. We got a little bit of energy, which means you can keep running. You didn't have to fall over after 15 seconds of maximal exertion. Now what? Well, in an ideal sense, in a perfect world, you are jogging at a reasonable pace, in which case you can go from pyruvate, which is step 1 of cellular respiration, and take it to the mitochondria, where you then go through steps 2 and 3, which we'll get to in a second. But sometimes we're sprinting, and pyruvate then starts to build up, right? So if I'm running at top speed, I can't keep this up for long because I broke glucose in half to pyruvate. I get a little bit of energy. That's good because that's what's allowing me to keep running. But pyruvate then becomes an acid. That's bad. Acid is building up in my tissues. As I change the pH of my tissues, my muscle proteins start to change their shape. Now nothing works. This is a cause of, say, a cramp, et cetera, and I'll get back to that. Now, I don't want pyruvate, so one of the things I do is I convert that into lactic acid, which is better. Lactic acid is better than accumulating pyruvate, but still, this is not something that I want to continue. Okay, so building up lactic acid is bad, and you know this because let's say you've been sprinting and all of a sudden your muscles burn. All right, why do they burn? Well, because of the lactic acid which has built up there. If you've been lifting something, helping your friend move, and your arms are tired, they're fatigued, or you're fatigued after walking up a flight of steps, that's lactic acid. If your muscle is sore the next day, one of the reasons is lactic acid. So lactic acid can cause 3 things. It can cause fatigue, it can cause muscle cramping, it can cause your muscles to be sore the next day. Now, there are also a couple of other causes. So lactic acid isn't the only cause of fatigue. So I can run out of glucose, all right? So like maybe I haven't eaten So, there's a couple of causes. Fatigue could also then just be more of a psychological phenomenon, that just emotionally I've dealt with this problem too full. I just feel fatigued. I need to go on vacation, and then I feel fresher. So fatigue can apply to a couple of things. Lactic acid is one cause. Cramping can be caused by a couple of things. So lactic acid in the tissues, but also I've been sweating away too many ions. I can get a cramp because ions are needed to conduct electricity. So if I'm running a marathon, if it's a good marathon that has spent your entrance money wisely, then at a certain mile, it's not just water that you can pick up as you go jogging by. It's also Gatorade. Why? Because as I sweat, I'm sweating away water and ions. If I'm just replacing with water, then after a certain point, I've lost too many ions, and now I don't have the right ions to conduct electricity, and now muscles can cramp. If I don't, if I'm just needing ATP at a far faster pace than I'm generating it, the myosin heads can pivot, but they don't have ATP to detach and return. So muscle proteins are binding and they can't detach. So once again, there's a couple causes of cramping, but lactic acid is one of them. And then finally, there's a couple of causes that muscles can be sore the next day. I mean, including what's called micro damage, where you can actually pull collagen fibers. So the collagen fibers of a tendon or the paramycium are displaced. Z discs can be displaced. So there can be micro damage to a muscle. So I'm not saying that lactic acid is everything, the only cause of fatigue or cramps or why your muscles are sore the next day, but it contributes to all of them. So if I'm doing glycolysis and stopping, I break glucose in half and I stop. I don't do steps 2 and 3 of cellular respiration. Then lactic acid builds up, and that's bad. That can cause cramps, it can cause fatigue, it can cause my muscles to be sore the next day. Now, my liver wants to help me out. As we'll see in Anatomy and Physiology 2, the liver does lots of things. So one of the things my liver can do is it can take lactic acid and put it back into glucose. That's what's called gluconeogenesis. Neo means new, genesis means to make. I can then make new glucose by taking lactic acid from exercise and then putting it back to make new glucose. So my liver can help me out in this thing. Now, sometimes as I put lactic acid into my blood and it goes to my liver, then my liver then is one of the first places that builds up lactic acid. And that could actually be a problem. I could get like a side stitch. Maybe you've been running and that's, concerned you. You're like, hey, my arms are moving, my legs are moving, I'm breathing heavy. Why do I have this stitch in my side? That's because I'm shipping lactic acid to my liver, and it's an early place that's accumulating enough of this acid, which is really disturbing homeostasis. So good news about glycolysis, I am getting more ATP so I can keep on running, which is better than falling over when something with sharp teeth is chasing me. Bad news is if that's what I do and I stop, I don't do steps 2 and 3 as we will in just a second, then lactic acid builds up, which means my muscles are tired, they're burning. So I physically can't keep sprinting. It just hurts too badly after a while. The fatigue, the muscle burn. I could get a cramp. My muscles can be sore the next day. So lactic acid building up, that's a problem. Okay. The good news is I don't have to stop. All right. So as once again, if you were going to study this more profoundly, you can see that you can then take the pyruvate to the part of your cell called the mitochondria. And here we have steps 2 and 3 of cellular respiration. All right. So in what's called the Krebs cycle, we take pyruvate, which is a 3-carbon molecule, and we break it down over a series of steps. This is occurring only in the mitochondria. So pyruvate, all right, which is this 3-carbon molecule, then gets converted to carbon dioxide in a series of steps. All right. I think that's right. So it's here. Okay, I'm sorry. I told you to go here. That's Um, here we are in the Krebs cycle. So notice that this carbon, the black dots, that came from whatever food you ate, like the French fry or the potato chip. And what happens in a Krebs cycle is, this could be a really long story, but the really short story is it gets converted to carbon dioxide. So notice that this came from sugar, but now look, this one carbon, that's a carbon dioxide. And then as we add it to a starter molecule and go around the circle, we will then add it to more carbon dioxide, which then comes out your nose. All right, so we added 3 carbons from our the sugar in our diet. There's one carbon dioxide, there's another carbon dioxide, there's another carbon dioxide. And now when I'm exercising, I'm exhaling that one, and that one. So it came into my body as sugar. It is now leaving my body as carbon dioxide. Now, the big piece here is if glucose is carbon, oxygen, and hydrogen. Once carbon dioxide comes out my nose, I'm obviously getting rid of the carbon and the oxygen. But what happens to the hydrogen? Well, the hydrogen, it goes to the inner mitochondrial membrane. It gets passed along a series of electron carriers in such a way that the positive part of the hydrogen atom, the proton, is in this space, while the negative electrons are in that space. So we take the hydrogen from the sugar that we eat, we pass it from carrier to carrier to carrier in what's called the electron transport system, in such a way that the hydrogens are out here, the electrons are there. That's not a happy situation. The hydrogens want to go to where the electrons are. And we say, we'll let you, hydrogens, if you want to go to where the electrons are, we will let you on one condition. You have to go through this special door. This special door, which will let the hydrogens go from the intermembrane space to the matrix in the mitochondria, has ADP and P weighting. And when hydrogen comes through, then ADP plus P, they join to create ATP. So as hydrogen from the food you eat gets reunited, the 2 separate halves, we generate ATP. All right, so this last phosphate gets added here. And this is how the food you eat is going to produce most of the ATP in your body. So I know some of you are saying, oh, this is too complicated. Well, okay, fair enough. But you run on energy. All right, you need ATP to move, to think, to keep warm, to grow, to maintain your tissues. This is where most of it's coming from. So even though it's a little hard to understand, because this is essentially the energy budget for your entire life, you know, at some point biologically, it's worth taking the effort to understand it. And so the hydrogen from the food you eat gets reunited. And in the process of going through this gate, we generate ATP. Now, hydrogen has to be bound to something to make it stable. So I'm inhaling oxygen. And this is why, if I want this next part to occur, if I don't want to build up lactic acid, if I want to break pyruvate down completely and get a whole lot of energy, the only way that works is if I'm getting oxygen. So this part of the process, mitochondrial respiration, requires oxygen, unlike glycolysis. And so as a result, if I'm sprinting, my AT demands are so high that I can't get oxygen to my muscles fast enough. I build up lactic acid, which means I fatigue. But look at me now, I'm not sprinting. I'm walking at a leisurely pace. As my muscles require ATP, because I'm getting oxygen to my muscles, I don't have to stop at glycolysis, step 1. I can now go to mitochondrial respiration, where instead of making 2 ATPs and building up lactic acid, I'm making 36 ATPs and now producing carbon dioxide and water. Carbon dioxide can come out my nose, water I can get rid of a couple of ways, and so I'm not building up lactic acid. So when I exercise, instead of doing glycolysis and then stopping, which then builds up lactic acid, which can cause fatigues, cramps, and muscle soreness. Here, I'm doing all of it, doing mitochondrial respiration, which means that now I can keep on going. So I can, if I'm swimming slowly, if I'm walking slowly, if I'm walking upstairs slowly, I don't have to stop, all right, because of fatigue and muscle burn, all right? So that's how I use glucose as my third energy source. Now, just one little quick point before I get back to the order of energy sources. Let's say I sprint and I run this race, all right? So obviously in the middle of my race, you can understand that, I'm breathing heavily, all right? But now let's say I stop, the race is over. And then what do I do? All right, once I stop exercising, I'm still, which is just weird. Why? Because I've stopped running. I'm no longer exercising. I'm just sitting there. Why am I still sweating? Why am I still breathing heavily? And the answer is because now I have this oxygen debt, okay? That my muscles have not gone back to their original state. The original state, I had ATP, I had creatine phosphate, I had oxygen bound to myoglobin in my muscle, I had no lactic acid. That's a resting state. But once my race is done, I'm not at the resting state, all right? So I need to keep breaking down sugar to make more ATP stores to get back to normal. So at rest, I have ATP just in case I need to sprint if like a wolf appears out of nowhere, all right? Well, I need to generate more ATP because it's all gone now. I need to generate more creatine phosphate because it's all gone now. So even though my race is done, I need to make more ATP and creatine phosphate to get back to normal. So at the end of my race, I'm breathing in oxygen so that I can break down sugar to produce ATP and creatine phosphate, getting me back to normal. Also, my muscles are full of lactic acid that build up during the race. That has to go away. The lactic acid built up because I didn't have enough oxygen. And so now I need oxygen to get rid of the lactic acid, all right? And so even though my exercise is done, I still have this oxygen debt. I'm going to undergo what's called recovery oxygen uptake to get back to resting levels of ATP and creatine phosphate to break down lactic acid. Also at rest, I have oxygen bound to myoglobin. That's that version of the globin, just like hemoglobin binds oxygen in blood, myoglobin binds oxygen in muscle, making it red. Hemoglobin makes blood looks red, myoglobin makes muscle look red. I have oxygen bound to myoglobin. So now I'm continuing to breathe heavily so that all of this can happen. Why am I sweating? And the answer, once again, there's a longer answer to this, but the short answer is in the universe, whenever energy changes hands, heat is generated as a waste. When you touch a light bulb, it's hot. When you touch your car's hood after it's been running, it's hot. Why? We didn't want that. We wanted the light bulb to generate light, not heat. We wanted the car to generate kinetic energy forward, not heat. But the answer is whenever I do anything in this universe where there's an energy transaction, heat is generated as a waste. So now as I'm burning up sugar, all right, in this oxygen debt phase, I'm generating heat. I don't want to, but it's a law of physics that all of these chemical reactions, breaking down sugar to make lactic acid, to make pyruvate, breaking down pyruvate to make carbon dioxide and water, that's now generating heat. So even though my race is done, I am still performing chemical reactions which are generating heat, which means I still have to sweat to lose this heat. So even though my exercise is done, I'm still sweating and breathing heavily. One last thing, however, I'm a mammal. And as a mammal, I'm 100 degrees all the time, even in winter, even when it's colder than 100 degrees. And this is why my muscle contractions generate heat. So the way I keep warm is when it's cold outside, I tell my muscles, hey, muscles, contract a little bit. Heck, even shiver if you have to. If not, then just contract a little bit. And so that then allows me to generate the heat, which keeps me warm. Okay, so all of these are fascinating, but we're almost done this one major topic. When I need ATP, where do I get my ATP? Well, first off, my muscles have ATP stores. Secondly, I can use creatine phosphate. Third, I can use glucose. I can break glucose down in glycolysis, or if I have enough oxygen, mitochondrial respiration. Fourth, when my glucose runs out, I can use fat as an energy store. All right, so lipids are my fourth energy store. Now, even though my body has lipid in it, it will use glucose first. So I use glucose first. I only start to use triglycerides in my fat stores when my glucose runs out. So this might happen at night, all right? So if it's been a while since I've eaten, now in what's called the post-absorptive state that you'll study in AMP2, I start to use lipids as my fourth energy source. If I'm running a marathon, and I've run a couple, marathoners know, that maybe around mile 18, mile 20, what do you hit? You hit the wall. If you don't know what I'm talking about, you haven't run a marathon. All of a sudden, you were going along well enough, but now every step is painful. You just hit this wall, and all of a sudden, just like life is horrible. And then actually you'll get through it, and then you can keep on jogging. What happened at the wall? Then at the wall, I ran out of sugar. All right, even though I've been eating spaghetti, and bulking up on carbs for the past couple of nights, I'm out of sugar, and now I'm going to my fourth energy source of lipids, triglycerides, and that's stressful. So then I can use as my fourth energy source triglycerides. Now, what happens if you're out of fat? Well, then you can use your fifth energy source in your body. You can use protein, but this means you're starving. Obviously, you don't want to use protein as an energy source because, to power muscles, because muscles are made of protein. And so if you were to look at energy sources in exercise, ATP first, creatine phosphate, glucose. Once you're out of glucose, you can then use triglycerides, and then proteins. Now, triglycerides and proteins can use the liver and a bit more complex metabolic reactions, which AMP2 will cover, but those are the energy sources of your body in order. One other quick thing before I get to, as we start to run out on topics, we have muscle tone, all right? Even when my muscle is at rest. Right now, my biceps brachii isn't contracting. I'm not flexing my forearm. It's at rest. There are some muscle contractions going on in there. Obviously, it's not flaccid. And so I do have some sort of tone. Even a muscle at rest has some muscle cells contracting. And this is because I'm using, among other things, muscle contraction to keep me warm. All right, it's not 98.6 degrees out. So to maintain a body temperature of 98.6, I have to have muscles contract, and I use that waste heat to help keep me at 98.6. So I do have some tone at all times. So there's just that. If I exercise, then that will increase the rate of muscle contractions in those muscles. So my muscle tone increases. I usually joke, that if you're going to, weight lift once a week, students, when would you do it? And then some will say, well, Friday afternoon. Why? Because Friday night you're going out to a party, and if there's one time in a week where you want to, appear that you have good muscle tone to impress those at the party, it's going to be Friday night. So you would then weight lift, and now the background state muscle contractions, your muscle tone will increase. So exercise can increase this. But as in the healthcare professions, we also look at muscle tone, because if muscle tone is too little, so if like I have no muscle tone here, the muscle is flaccid. If this muscle is flaccid, it could mean a neural problem. All right, it could mean ion imbalances. It could mean I had a stroke. All right, so muscles with too little tone can be a problem. Muscles with too high a tone can be a problem. A spastic muscle, once again, could be indicative of a problem. So muscle tone is not just something, you know, to worry about with exercise and I want to look good in the gym or whatever. You know, it also has applications to neurological issues and then, you know, hormone imbalances which change ion levels. Okay, as we start to wind down the topics in this chapter, it then gets pointed out that I've been talking about muscle as if it's one thing. It's not, because there are different kinds of skeletal muscle. We know this from when we, you know, buy chicken in the grocery store. You know, you can pick, you know, white meat or dark meat. Now, meat is muscle, but then obviously in the chicken, it comes in colors. All right, so you know why is there these two different kinds of muscle? Well, even though, you know, we aren't quite as dramatic as the chicken, we still do have what are called fast glycolytic fibers, fast oxidative fibers, and slow oxidative fibers. There's different kinds of skeletal muscle. Not only does the chicken have it, but we do as well. So, the fast glycolytic fibers, they tend to be wider, all right? That's why the chest muscles of a chicken, you know, that's so much muscle. And we can have these, you know, also in our arms, our chest. These can be larger in size. They primarily use glycolysis for their ATP source. Slow oxidative fibers, they're narrower, and they tend to use cellular respiration as their ATP source. Now, if you think of chickens for a second, chickens don't fly south for the winter. So when they use their wings, it's not for endurance flying of long distances. Chickens only use their wings for a quick burst of activity to jump up into the branches of the tree so they can get away from a predator. So if they're going to use their wings, it's in a short burst of energy, not a long burst. And so if I was going to be using fast glycolytic fibers, since glycolysis is the main source of energy, it's for a fast burst. Like in my arm muscles, I have these. That's why if you exercise your arms, they'll get big. All right, you'll increase the size of these fast glycolytic fibers, but they'll fatigue easily. Right, so I'll lift something heavy, helping my friend move their couch, but my arms get tired. All right, if you're a marathon runner, you're exercising all the time, but your legs don't get big. Marathon runners, they don't have big legs the way that a weightlifter would have big arms. All right, because here the marathon runner is relying on slow oxidative fibers, not glycolysis. If you're using glycolysis while you're running, it's because you're sprinting and you'll get tired easily. The marathon runners, they want cellular respiration, so that they want to use oxygen so that lactic acid doesn't build up. Now we actually have fast oxidative fibers which are actually in the middle using both glycolysis and respiration. And so if you look at, say, a sprinter versus a marathon runner, let's say really good ones, our sprinter would tend to have more of these fibers, all right, in their legs. A marathon runner would tend to have more of these fibers in their legs. Some of that can be modified through your type of training, but some of them is genetic as well. You might just, be naturally better at sprinting versus like distance running. Back in the day, when I was, cross country teams, I ended up being, I ran marathons, but I was never a good sprinter. Everyone else on the team was far better at, there were, individuals, including my brother, far better at sprinting. But I ended up running marathons, while some of them, that was just not something that they, took to. And so some of this is how you train, but then part of this is just the natural variation. Our hair color is different, our blood types are different. The ratio of fast oxidative fibers to slow oxidative fibers are different. Now, when you see this in the supermarket in chickens, this meat looks darker. All right, so chickens are on their legs all day long. They have slow oxidative fibers there. Their leg muscles can't afford to fatigue the way that their chest muscles do. And the reason that these are darker in color is they have lots of myoglobin. just like hemoglobin makes blood look dark red, myoglobin, which binds oxygen in muscle, makes muscle look dark red. It also has more capillaries and more mitochondria to break down sugars using oxygen. So the capillaries, the myoglobin, are giving this muscle a darker color. So you can distinguish in the supermarket between the slow oxidative fibers and, say, the fast glycolytic fibers of the chest muscles. So here, these look lighter in color. They have less myoglobin, less in the way of capillaries. And so we have these three different kinds of muscle, which a lot of people who study anatomy and physiology, you might be going into sports medicine or exercise physiology. And so we can then, talk about these. We can talk about how they are recruited during exercise. Our fast glycolytic fibers give us short, intense bursts of activity. Once again, you're sprinting, you're lifting something heavy, but these fatigue easily. These slow oxidative fibers, this is what we use for endurance activities. So like walking or just maintaining posture. When we talk about exercise, as once again, many people are interested in, intense workouts using these fibers cause them to get more myofibrils, more sarcomeres. So this causes them to hypertrophy. That's why, certain exercises would then give you bigger arms or bigger chest muscles. You don't want that if you're, running long distance, because these big muscles would actually, when they contract, that would, squeeze blood capillaries and stop blood flow. That's not what you want in a long distance race. It might help you lift a heavy weight in a short burst of activity, but not a long burst of activity. And so if you were, studying anatomy and physiology to get more into exercise physiology, then, the different types of training regimes, which affect, fast oxidative fibers or slow oxidative fibers would then obviously be of interest. One last point is, if we were to hurt ourselves, skeletal muscle actually does heal a little bit. Now, the way we made skeletal muscle in as embryos is we had lots of little cells called myoblasts fused together to form these huge cells with lots of nuclei. And in muscle tissue, we still do have what are called satellite cells. And satellite cells, to a limited degree, can fuse and then make these additional skeletal muscle cells. So if I were to injure myself, I could actually get a little bit of skeletal muscle. I could make more cells. Now, not as good as smooth muscle, how it regenerates itself, but cardiac muscle doesn't regenerate at all. So skeletal muscle is a little in the middle there. So I can injure it, but then also skeletal muscle, if I'm not sending it signals. So for example, if I have a stroke and I'm no longer sending commands to this muscle or this muscle, it will undergo atrophy. The skeletal muscle cells will become smaller and thinner, but at a certain point they may die. And then at a certain point, that damage then is permanent. So skeletal muscle does have a little bit of an ability to regenerate, but not much. And so in an anatomy and physiology course, the chapter on muscle tissue is long, and I've broken this into two videos. The first video went through with the anatomy and the sliding filament theory. This video is attempted to go through the second half of the chapter and all remaining topics. Some topics, such as cellular respiration, I cover in greater detail elsewhere.